What are these? Oh yeah, that's eczema. I have it, so you can have it too.
Huh?
That was an example of a genetic disease. Your DNA can pick up mutations when a new copy is made. Some are harmless, others not so much. Think of it like copying a sentence. You accidentally write the mat was sleeping. Normally, we have systems in our cells that fix these mistakes like DNA polymerase and exonuclease. But with inherited diseases, the mistake is already built in, and your body considers that the correct instruction. And unfortunately, these genetic diseases were considered uncurable. But here's where CRISPR comes in. Clustered, regularly interspaced, short penetrating repeats, which can fix the mistake for you. Gene therapy is one of the most promising breakthroughs in medicine today. Scientists start by identifying the faulty gene, then they design a guide RNA complementary to the mutation that works like a GPS, guiding the Cas9 protein to the exact spot in the DNA that needs to be cut. To get this gRNA into your cells, scientists often use harmless viruses, kind of like overrides through a method called homology-directed repair. What? Uber cancel my ride? At least this ride is reliable. After Cas9 cuts the faulty gene, the cell tries to repair it, but it often messes up, so the gene gets permanently turned off. But how about addition or modification? Scientists will then proceed to make the healthy gene, with homology arms, which are bases before and after the correct gene that match the stretch of bases before and after the shutdown gene. These guide the cell where to insert or replace the DNA, so the cell recognizes the homology arms, lines up the new gene perfectly, and inserts it exactly where it belongs. This kind of precision editing, it could change lives for people with sickle cell disease, cystic fibrosis, and even some cancers. Dad, it's curable!